Let's begin with another session for today on role of telomerase and telomerase in aging. We have distinguished speaker for today's session, Mrs. Sahaya Nadar ma'am. Mrs. Sahaya Nadar ma'am has completed her MPharm from Siusha College of Pharmacy in the year 2014 with the second rank is in the SNDT University. She has completed her B.Pharm from St. John's Institute of Pharmacy and Research. She has total 8 years of teaching experience. She was awarded the Ratan Tata Scholarship for Academic Excellence in the year 2009. She has 12 reviews and 4 research articles in international and national journals to her credit. She is working as an active reviewer of Bentham journals like anti-inflammatory and anti-allergic agents in medical chemistry, anti-cancer agents in medical chemistry, combination chemistry, and high throughput screening, and Taylor and Francis Journal, SAR and QSAR in environmental research. Her area of expertise is computer-aided drug design for synthetic anti-infective and anti-cancer agent. With this brief introduction, I now request ma'am to take over the session.
Thank you, Rennie, for the introduction. Is my screen visible? Yeah. So good morning, students. Today we are going to explore a new topic, role of telomers and telomerase in aging. This will be the outline of my presentation today. We will try to understand what are telomers, what are the result of telomers dysfunction, factors that affects the aging process, telomers and its association with aging, what is the role of this enzyme telomerase, what are the methods to measure telomere length, what are the future trends and references. Talking about telomers. In human, the DNA that we have in our cell has a repetitive sequence at the end of telomers. So these telomers are basically repetitive DNA sequence at the end of human chromosomes. They contain thousands of repeats of 6 nucleotide sequence, that is thymine, adenine, and guanine. In human cells, there are 46 chromosomes and hence 92 telomers, one at each end of the chromosome. So think of it like this. Telomers effectively cap the end of your chromosome in a manner very similar to the way a plastic on the end of your shoelace caps and protects the shoelace from unveiling. Similarly, as you see in the image, your shoelace being protected by the plastic cover, similarly your DNA or the chromosome, at the end of the chromosome, you have these telomers which protect your chromosomes from getting unveiled. So this image is a fluorescent stained chromosome which is shown in red on a microscopic slide, and what you see in yellow as fluorescence is the telomers which sits at the end of each of your chromosomes. So if there is a telomere shortening or telomere dysfunctioning, what are the different consequences that can happen? It can lead to cytogenic abnormalities, like when your telomers are shortened or there is a dysfunction, 2 of the ends of the chromosome can fuse together, called as telomeric fusion. This fusion can lead to formation of bridged chromosomes. These bridged chromosomes ultimately leads to breakage of the chromosome or missegregation. All of these leads to fusion of these small chromosome units. These small chromosome units can lead to various kinds of cytogenic abnormalities. In the year 2009, for the discovery of how chromosomes are protected by telomeres and the role of the enzyme telomerase, the Nobel Prize in Physiology or Medicine was awarded to Elizabeth Balker, Carol Gerard, and Jack Szostak. So what is the connection of telomeres and aging? Is there any connection? Talking about aging, there are various factors that are involved in the process of aging. One of the factors that we are going to discuss today is the telomere length or the telomere shortening. Before we move to that topic, what are the other factors that are affected or the factors that are affecting the aging process? Now, as all of us know, aging is a process which is a chronological aging process. Nobody can stop the process. With years passing by, our age increases. So that is a chronological aging factor, and the risk increases with increasing time. The next factor, very key factor, which affects the process of aging is oxidative stress. Oxidative stress happens because of formation of free radicals. These free radicals ultimately damage our DNA, the proteins, and the lipids. The other factor that affects the process of aging is glycation. Now, glycation happens because of the normal glucose. Glucose is a source of energy. This glucose, when it goes and binds to the DNA, protein, or lipid, it inhibits the various pivotal functions that these have in our body. So all of these factors affect the process of aging. What we are going to discuss is how the telomere shortening ultimately affects the aging process. So when I speak about telomere shortening, this is the length of the telomere which is present at the end of your chromosome gets shortened over a period of time, and ultimately it leads to the process of aging. So to understand the role of telomeres, let us first see how the DNA replication happens in our body. So when your DNA replicates during cell division, a small part of your RNA, a small piece of the RNA, which is called as a primer, helps to get the process of this replication started. So once a primer attaches, cellular machinery can copy the DNA strand. So whatever DNA strand is there, as soon as the primer gets attached to this DNA strand, your DNA synthesis process comes. Okay. Since the primer, if you look at the image carefully, since the primer is not attaching itself to the very end of the DNA strand, the copying that happens, the process of copying that happens is missing the end section of your DNA, right? So there is a missing segment in the copy of the DNA strand that is formed. So next time when your cell divides, the copied DNA loses more end of this section. So with with increase in the cell division process, at the end of each cell DNA replication process, the strand becomes shorter and shorter. So in humans, about 50 to 250 base pair are lost per cell division. Now, what is the role of telomeres and how does it help your cell to keep the DNA intact? So telomeres are repeated sequence, as we discussed, at the end of your DNA strand. They help to protect the DNA strand from getting shorter during every cell division process. So this is your normal DNA strand, as we saw in your first case. Here, the DNA strand is shown with the telomere. So ideally, the DNA strand has a telomere unit at the end. Now, when the primer attaches and the replication happens, the copy of the DNA strand is formed. It is, of course, shorter than the earlier copy, but because of the presence of telomere, the copied DNA strand is much more bigger than the one which was synthesized without the telomere strand. All right. So in germline cells like your egg and the sperm, there is an enzyme called as telomerase, which is responsible for adding more repeated sequence to the end of the DNA, thus making these cells basically immortal. So your germline cells like egg and sperm are usually immortal in nature because of the presence of the enzyme telomerase. But in the somatic cells, the telomerase enzyme function is very low. Okay, the enzyme functions, the enzyme is present, but the level is quite low, which makes this cell ultimately mortal. Okay, so when the cell division happens, when there is a DNA replication taking place in a somatic cells, At a point when there is no telomeres left, the cell is at a very critical level and the cell can no longer divide, which leads to the process of aging. So why do your telomeres shorten each time a cell divides? There is an image that is put here. Think of yourself that you are standing at the corner of a room and you're trying to paint the entire room. Okay, at the end you are unable to paint the corner, so that corner end is left. It is as good as that. Before a cell can divide, it makes copies of its chromosomes so that both the new cells will have identical genetic material. Okay, to be copied, a chromosome's 2 DNA strand must unbind, and the enzyme DNA polymerase then reads the existing strand to bring the new strand. Now it begins the process with the help of the short pieces of RNA, which we had already discussed, which are called as primers. Each new matching strand is complete. It is a bit shorter than the original strand because of the room that is needed at the end for the small piece of RNA to attach. So as we age, our length of the telomere keeps on changing. So if you look at a newborn human, telomeres are approximately 15 to 20 kilobyte in length, and they ultimately shorten gradually throughout your age, suggesting that the telomere length may serve as a surrogate marker for aging process. So telomeres and aging. Once the telomeres shrink to a certain level, the cell can no longer divide. Its metabolism slows down, it ages, and the cell dies. So how is this telomere length and aging actually related? At the birth, the average telomere length is 10,000 base pairs. As you age, at around 20 years of age, approximately, the average telomere length is 8,000. By the age of 50, it gradually decreases to 7,000. And so it goes on losing 50 to 100 base pairs a year until only 4,000 remains at the age of 100. Now, this telomere length is varying cell to cell. Okay, for example, the graph that we have depicted is of human blood cell. So if you see the telomere length in your blood cell, it is varying from the data which is put on the slide. The slide data is the average taken for all the cells present in your body. Moving to the enzyme telomerase. Telomerase, which is also called as the fountain of youth, is known as human telomerase reverse transcriptase, an enzyme that adds telomeres back on the end of the chromosome. So as your DNA replicates, the telomere length also shrinks, right? What is the role of this enzyme telomerase is, telomerase adds your telomere back to the end of your chromosome. So how does this telomerase work? So telomerase places one strand of DNA on the RNA, positioning itself so that the template lies adjacent to the tip of the chromosome. When this enzyme adds one DNA nucleotide at a time, ultimately it keeps on adding until a full telomeric subunit is formed. When the subunit is complete, the telomerase can attach another by sliding to the new end of the chromosome and repeating the synthetic process. So this is how it works. The telomerase enzyme, which is shown in green, it binds to the 3' flanking unit or flanking end of your telomere that is complementary to the telomere RNA. Okay, so this is your telomere RNA and this is the 3' flanking unit. So your telomerase enzyme basically binds to the 3' flanking unit, which is complementary to the telomerase RNA. Bases are added using the RNA as a template. So base pairs are getting added and the telomerase then relocates. After the base pairs are added, it gets relocated. And this step, that is the step 2, where the base pairs are getting added, keeps on getting repeated. So the DNA polymerase complements the lagging strand. So that is how the entire process takes place and the telomere gets added back to your DNA. The next question that arises in your mind is, can you measure your telomere length or the length of the telomere as it is associated with aging? Yes, of course. There are a number of different ways in which the telomere length can be measured, all but with varying degree of accuracy and different requirements. Few of the techniques that we will be seeing were ultimately pulled out because of the varying degree of accuracy. So telomere length measurement. If you measure the length of your telomere, what are the benefits that you can acquire? So the primary benefit of telomere test may be the information that it provides to an individual regarding their biological age. So it is not only limited to the biological age. With aging, we also acquire different diseases, degenerative diseases, and ultimately it is also associated with the cell aging process. Okay, it is not only about the aging, we are talking about the cell aging process. One of the method which is very popularly used is quantitative polymerase chain reaction. Now this quantitative polymerase chain reaction is a 30-40 steps cyclic reaction wherein your DNA is extracted. Okay, we are not going to look into the detailed process, but I will just give you an overview. The DNA is extracted. It is denaturation of the DNA takes place, okay, which is for 1 minute at 94 degrees Celsius. The second step is annealing, where the forward and reverse primers, again the step is of a 45-second step, and then there is an extension step. So this process, polymerase chain reaction, quantitative polymerase chain reaction, is continued for 30 to 40 cycles, after which the length of the RNA, the length of your telomerase is estimated. One of the very popular method in the earlier 2000s in the UK and the US was the at-home telomere measurement kit. Okay, so TeloYears was launched in the earlier 2000 in the UK. This was a telomere measurement kit which was available in the market to measure the telomere length, which will tell you what is the your actual age, not the year that you say I'm 30 years old or 20 years old, the actual age of your body that was predicted by this TeloYears kit. But there was a drawback of this kit. It was not accurate. People started claiming that a 30-year person who is going to check his years with a TeloYear kit was shown that he is 80 years old. That is the cells that are there in the body are getting older and older. So people became very panicked. They started panicking. So this kit that was there was ultimately banned. And there were more techniques that came in which were helping to accurately determine the telomere length. So benefits of telomere test. It is a marker for biological age. This information may enable a person to make adjustment in their lifestyle that can lead to a better health and a longer life. So telomere shortening is related to age, degenerative disease, cardiovascular disease, stress, and causes impaired immune function that might also increase the cancer susceptibility. So if you could predict your telomere length, you could make lifestyle adjustments to just prolong your life. What are the recent trends? So in the year 1997, a company called as Geron received a patent relating to RNA component of this human telomerase enzyme, and they believe that while combining the RNA component with the telomerase gene, product that could immortalize cell without turning them carcinogenic could be designed. Okay, there are various products that are already available in the market. So if this technique works, not only would the possibility of rejuvenating human cells to treat age-related disease be more feasible, but also other techniques that are currently limited by mortality of human cells could happen. So Telomere Guard is again a product which was launched. TA65. Now TA65 is produced at a very low level in astragalus plant, but the company that is there, TA65, they claim that they purify and concentrate the substance which is thought to turn on the enzyme telomerase that acts to maintain or lengthen your telomeres. Okay, so telomerase is usually off in adult cell, as we had discussed. In somatic cells, they do not, after a period of time, the enzyme telomerase is not that active, but it is active in your cells, immune cells, eggs and sperms, and in malignant cancer forming cells also the level is quite high. Okay, so the TA65 pill that was formulated by this TA Science company went on to the market without any FDA approval because they claim that it is just a supplement and it is not a drug. There is another product B, which is product B antioxidant plus telomere support. This helps maintain youthful cells. These are all claims by the company. Okay, they claim that it helps to maintain youthful cell function and healthy telomeres. They say that it helps to protect them from harmful free radical that can accelerate aging and poor health, targets the source of accelerated aging, that is oxidative stress, and it also regains and maintains youthful aging. Another company that has evolved in the UK, Telomers. Telomers has collaborated with LifeLength. LifeLength is a pioneer in telomere length measurement. Okay, and they have all these accredited labs, clinical labs set in the UK. Now, LifeLength has developed a unique proprietary high throughput technology, which is helpful to measure the telomere analysis or measure the telomere length. Okay, this technology is called as TAT, with telomere analysis technology, which helps to monitor or measure the telomere length. To conclude, if you could use telomerase to immortalize human cell, we may be able to mass produce cells for transplantation, including insulin producing cells to cure diabetes, muscle cells for treating muscular dystrophy, cartilage cell for certain kinds of arthritis, and skin cell for healing severe burns and wounds. So not only is it related to aging process, but if you could immortalize human cell, it would ultimately help to cure a lot of human problems. An unlimited supply of normal human cell grown in the laboratory would also help efforts to test new drugs and gene therapy. Okay, to end, I would just like to say we all love to dream of eternal youth, but time will never stop. But we can try to make it go slow. So maybe then we can reverse the clock of life. These are few of my references. Thank you. And of course, man is the maker of his own happiness. We choose how we want to be. Okay, so with this, I will end my session. Any doubts?
Good morning, everyone. It's such an honor for me to get the opportunity to thank you all. I extend my gratitude to our distinguished speaker, Mrs. Sahaya Nadar ma'am, for giving us excellent coverage on the topic role of telomers and telomerase in aging. Ma'am has focused on various points like what are telomeres, cytogenic abnormalities from telomere shortening and telomere dysfunction. She also showed us about the few Nobel Prize winners, telomeres and aging and its connection. She also explained about the factors that results in aging and DNA replication process and telomere. She also has explained about why do telomeres get shorter each time a cell divides. She has also explained about telomere length changes in human aging, brief about telomere and aging, telomere length and aging. She also has given us idea about the at-home telomere length measurement kit that was launched in UK that shows us the actual age of body. She also explained benefits of telomere test about the various pills like TA65, product B, and antioxidant telomere support. And our sincere our sincere thanks to Mr. Albert D'Souza sir for his encouragement, and thanks to all the dignitaries of the support of the management. A special thanks to our principal, Dr. Savita Taru ma'am, for immense support. I would like to thank the coordinators for their guidance and support. We are all excited to meet you. See you all in the upcoming session of SCIENTIA. Thank you for being there. Have a great day with us. Feedback link will be provided in your respective WhatsApp group. Kindly fill it in. That will help us improve our program. Thank you.